Before I get started talking about scholarship and scholarly work, I would like to use this opportunity to express my gratitude. I have a long list of acknowledgement here. So first, I would like to thank colleagues, collaborators, and students who wrote letters of support for the application of this award. And I want to thank the selection committee for the work they put in, and I would like to thank Faculty Assembly for organizing and inviting me to give the presentation today. And I would also like to emphasize that this award is a recognition of the work of everybody on the team. Not just myself, but I have an amazing team that we work with. Students, faculty, and staff at the School of Engineering and Technology. Some of them are here today. And in particular, I would like to thank Ling Hong Hong, a research scientist in my group, who has played a critical role in my research program since 2014. And I would like also to thank Wes Lloyd, assistant professor at the School of Engineering and Technology, for inspiring us to pursue research opportunities in cloud computing. I learned all my cloud stuff from Wes, so that was amazing. And finally, I would like to dedicate this award to my family and in memory of my father. So now with that note, so these photos show students in our group over the years. So some of you in the audience today would probably see your photos here. And that's, so an overview of what the presentation today. So first of all, I know the title of the presentation sounds a little bit technical, but I would make it not so technical. So I would start by giving a quick introduction to bioinformatics so that we'll all be on the same page. And I'll give an overview of the projects that we currently focus on. And then I would talk about what we mean by reproducible research and why we care about reproducible research. And then I'll talk about more specific projects that our group works on. So let's get started with a quick introduction to bioinformatics. So when I started graduate school, actually we don't call it bioinformatics. We used to call it computational biology. I don't know why that term has been fallen out of favor these days. So what is bioinformatics? And these days we just use these names interchangeably. So bioinformatics, according to Wikipedia, is an interdisciplinary field that develops methods and software tools for understanding biological data. So the way I look at it is a continuum between biology and computational sciences. Notice that I didn't say computer science, but computational sciences, which is much broader. So we include statistics, applied math, and many other disciplines as well, engineering, etc. So I myself started on this end of the spectrum. And after I got my PhD in computer science, I tried to walk towards the other end by spending more than 10 years at the Department of Microbiology in the School of Medicine in University of Washington, Seattle. And after that 10 years, I decided to take a zigzag and swing back to the other direction. So, but there are many bioinformaticians that start at the other end and walk towards the other side as well. So what is really cool and attractive about bioinformatics is the collaborative nature of how it's an interdisciplinary field and the opportunity to work with people who are different from myself and with different background, different perspectives, different training. So it's also typically the way it works is it's an iterative cycle. So we have our collaborators working in the lab generating data. And then when the data is generated, a lot of data processing, data handling needs to be done. And after we clean up the data, we would have to build mathematical models. We apply different machine learning methods, data science tools to analyze the data. But that's not the end. When we are done, it's very important to look back to our buddies who generated the data in the first place. That is how we actually draw biological insights and actually make a difference in a translational setting. And modern day biology is very much a data-rich science. Terabytes of high throughput data have been generated and being available in many public repositories. So there are many different types of high throughput data. Many of these types of data quantify the activity levels of tens of thousands of biological entities simultaneously. So biology in modern day is very much a data science problem. So that's where people like myself kicks in and could make ourselves valuable. And analyzing these big complex data could advance the understanding of biological mechanisms and diseases and facilitating therapeutic discoveries. That's what we want to accomplish at the end of the day, is make a difference in the well-being of people. So, and there are many different directions of bioinformatics research. So typically, there are many different types of research. One of them being most popular is the development of new computational methods. The way to get papers published is developing the methods is insufficient. We also need to show the methods that we develop is better than what's currently there in the literature. And so these types of evidence to showing that the methods actually draw useful insight could be in terms of validation in the lab, or it could be computational validation as well. So for example, if you build new predictive models using machine learning methods, and you have to show your model is more accurate or have higher recall or whatever quantity that you to extend compared to existing methods. And other types of research direction include generating and analyzing these new complex data sets, and how by analyzing these data, how the interpretation of the data would advance the understanding of diseases or biological mechanisms. And yet another direction of these types of research is to develop new software tools for the analysis of biomedical data. So my research, I have done all of these different directions at different stages of my career. So ever since I started at University of Washington, Tacoma, I focused more on developing software tools and expanded my comfort zone quite a bit. The idea is to match the interest of our students. Our students are mostly interested in practical applications that they could put on their resume. And also we could also, when in software development project, we could split up the projects into smaller chunks so that students would get more positive reinforcement and get publications in a relatively short period of time. So that leads me to the next slide, which is what we are currently working on, the kind of projects. So our goal is we are trying to make the latest technological advances in computer science, in container technology, in cloud computing. We want to make these advances accessible to biomedical scientists by developing easy-to-use tools to facilitate collaboration. So note that I highlighted easy to use using our Husky color. So the idea is that a lot of these technology are accessible to people who are trained in computer science, who are trained to use the computer to write code, to write program, to write software. But what we want is we want to reduce the barrier of collaboration so that after we develop the tools, it would be accessible to people who are not necessarily trained in computer science. So that's a big part of our work, is to make technology accessible. And there are some really cool characteristics of our work. First of all, we are trying to make our tools modular. So modern day, after we generate these big, complex biomedical data, there are usually a sequence of steps that you need to perform to analyze the data. And each of these steps could be performed using a different tool, different software that require a different computing environment. So by making our tools modular, is that if somebody comes up with a new tool for one of the steps tomorrow, you could immediately swap out what's currently in your workflow or your pipeline, your sequence of steps, and put in the state-of-the-art tool. So one of our characteristics is that we are trying to make our tool modular. And because of the nature of these big data, we want to leverage cloud computing so that not only we want to use cloud computing, we want to use it in a sophisticated manner. We want to use it in an intelligent manner so that our job would be able, it wouldn't cost you too much money because cloud computing is not free. And they usually charge you by how much time you use their resources. So we want the jobs to be run efficiently. We want to reduce the running time of the job, and we want to save money, save the users money. At the same time, we also want to make everything easy to use. And there are a zillion of different tools on various cloud vendors. And what we want to do is to make our tools accessible, is that we would help you to make some of these decisions. We would help you to turn the knob to the optimal setting, so you don't have to worry about all these knobs to turn and all these what's out there. So by using our tools, you can be assured that we have done the work for you. So that's what we are trying to do. And yet another major characteristic of our work is we use what we call software containers for each of the modules. The idea of this is that we want to enhance and facilitate the reproducibility of research. So at this point, I want to digress a little bit to talk about reproducibility, what that means and why we care about this. So reproducibility, there are a couple of ways we could look at reproducibility. One of the definitions for reproducibility is that when you are presenting your methods, your results, if you are giving sufficient details, so somebody else reading your paper, typically in the fine print in the method section, that the font size is 8 or below, like tiny, tiny print, that by reading that, you would be able to reproduce the work. If we have sufficient details, that the readers would be able to do that. And another definition of reproducibility is that if let's say we make the software that we produce as available in a public domain, if you could just run the software and get the same results as we did, as reported in the paper. So that's what we mean by reproducibility. And nowadays, a lot of the journals is in their editorial policy is critical to address the reproducibility of your work before it would be accepted. And it's also a big criteria of federal funding these days. So it affects how we get credits in the academic world, and it also affects how we get money in the academic world. So we should all worry about reproducibility. And also, so that our work would have the impact that we want. So in fact, there is growing alarm in the scientific community about results not being able to be reproduced. So this is an article in the Science magazine in 2015 about the cancer test. And actually, after this article, there has been many other articles in the Science magazine talking about issues and concerns about reproducibility. And the Cancer Reproducibility Project is an open science effort. And the goal is to replicate 50 high-impact cancer biology papers. So what they do is they are trying to test whether key findings in these papers that are published in high-impact federal journals, whether somebody else could independently reproduce the results. And the disappointment is that it turns out that it's not so easy. And the success rate of independent validation of the results is not very good. So, so that is definitely a problem. So this is documented in the literature. But what we have done is that we have also tried to do an exercise in a class that I teach. I typically teach a 500 level, a master's level bioinformatics class, and there are a couple of students in this room who have taken this class before. You guys are very lucky you didn't take this class 2 years ago. This is what I did to the students who took this class 2 years ago. So another thing that I really like to do in this class is I love crowdsourcing projects. So that is typically the final project in the class. So students would work on an open source project, and they would submit, and then we would see how we rank compared to everybody else in the world. And that is the way that we could have the impact outside our close community as well, in the entire scientific community. So in 2016, I had the class to participate in this particular crowdsourcing project. So in this project, it's called the Respiratory Viral Dream Challenge. So what they gave us are time series, some gene expression data. You can think about these as giant spreadsheets. And we have 2 different time points, and these data measure the activity levels of tens of thousands of genes and over time after they get infected by different viruses. So they were looking at 4 different viruses, H1N1, H3N2, RSV, and rhinovirus in 7 different studies. So the 2 time point is time 0, which is before infection. 24 hours is 24 hours after infection. So the goal of this challenge is to build predictive models using machine learning methods to try to predict who would get more likely to get sick after exposure to these different viruses. So this is motivated by the fact that, let's say we are all in the same room. If one of us is sick, don't tell me which one of you is sick. So sometimes we are all in the room, and some people would get sick after sitting next to somebody, but not all of us would get sick at the same time. So, so we want to predict what makes somebody more likely to get sick, even if we are all sitting together in the same room. If one of us is sick, it's not me. I can tell you that, not today. So, so in 2016, the students, they participated in crowdsourcing challenge, and they actually ranked very well. In one of the subtasks, we were actually ranked the top 10% in the world. So that was pretty amazing. And then the year after, I was thinking, hey, let's try to do a reproducibility exercise, because this is a public crowdsourcing challenge. So all the submissions from all the teams over the world are available from their website. So in 2017, I asked the students who took this class, it was quite unlucky for them, that to ask them to split up the work and try to reproduce the results from the submissions for the crowdsourcing challenge. So everybody have to do a couple of submissions. And they were not very happy about this task. But anyway, so the question I have for you is, how many do you think the students could reproduce? So this is a master's level class in computer science, and we are looking at about between 50 to 60 submissions. So I have about 20-ish students in the class, so each person takes care of about 2 submissions. So how many of these do you think we can reproduce? Any wild guess? 10. Anybody else? 3. You guys are way too optimistic. It's 0. None. Nil. Not even the ones we submitted ourselves. So I have to be honest to say. So all the challenges include a lot of the submissions have missing files. So for example, oftentimes after you download the data in the challenge, people typically massage them. They clean it up. They remove something they don't like. And then their code, their software, use the processed data. But nobody actually make the processed data available to other people. So we cannot, we just don't have the input data, so we cannot run the code. And some code was interesting. They just assume that you have access to their own personal Google Drive, which we don't. And then another issue is the environment setup. So oftentimes, like especially for these types of crowdsourcing challenges are usually marathons. So you start, like it takes many months. And in the process, when you are doing these crowdsourcing challenges in different stages, they release new version of software. And the new version of the software might not produce the same results as when you make the submission. So it's nobody's fault. It's just that you need to document the exact version you use. And sometimes they run out of memory or disk space. And some teams might have access to some supercomputers, very nice, that our students don't have. They're just running it on their laptop, so it might not work. And then there are also some code issues, like they assume certain software, certain tools that our students don't have. some of these tools that you might need to buy in order to use. And sometimes the code simply doesn't run, or sometimes people don't submit all the code. Like, let's say they have 5 different scripts, they just submit one of them. So that is also pretty common. So that was a very interesting exercise. Doesn't make people feel very good about this, but this is a very good exercise to actually very close to us. I say, hey, you know, even stuff we ourselves submit didn't run when it's the students from next year's class. So that was a very interesting exercise for us. So what we do is because we are a dry lab, we don't do any lab work in our group. We do only computational development. So when we look at reproducibility, we assume the data have already been generated. So we are trying to ask the question, what if we confine ourselves to look at the reproducibility of just the bioinformatics part, just the computational part, how easy or difficult it is to reproduce the results. And what we have just seen from the exercise in the class, that it is more difficult than you thought. So we have summarized some of the challenges in reproducing bioinformatics analysis results. One of them, as we talked about before, it is different versions of software and also software dependencies. Because oftentimes when people come up with new tools, they use some other tools in the process. So oftentimes we don't start from scratch, we build on top of what other people have done. So you assume when you are doing the analysis, you assume version X. And then, but when somebody else, when the paper got published, version X is not the latest version anymore. And when people use the next version, it might not produce the same results. So it might not be your software, it's somebody else down the chain. And those are oftentimes beyond your control. And multiple operating systems is an issue. And configuration and deployment. How do you configure your laptop? So again, a lot of these wonderful work were performed by students. I had a student was trying to reproduce some of the results. And what he discovered is that the language encoding of your laptop could make a difference when you sort different names. So if you use a Chinese version of the operating system versus if it's Australian English, English, American English, they could do the sorting slightly differently. And that is an issue if, let's say, if you have a G name that has a hyphen in there or some obscure characters, that could make a difference. And to make things even worse is that, as we talked about before, bioinformatics analysis is not one computational task. It's a sequence of many steps. So you have so many more opportunities to not be reproducible. If in any steps you don't get the same results as the published results, the end results is going to be not reproducible. And to make things even worse is a lot of the time, the software tools used in a biomedical community have a graphical user interface. So that it's not, well, to making things worse or better, it depends on who you're talking to. So the graphical user interface make it easier to use, but at the same time, you cannot run all these jobs in batch. You have to watch them, you have to click. If you are somebody like me, I automate everything if I need to do it 5 times. So what we do is we use software containers. So the idea of these software containers is that we don't distribute the code, we don't distribute the software, but we distribute the entire computing environment. So you can think about it as a software computer that runs on top of your actual computer. So there are many different types of software containers out there. We use Docker. So Docker containers wrap up a piece of software in a complete file system. So it contains everything you need to run. It has the code, it has the runtime, it has the libraries, it has the dependencies, everything. And this guarantees that it will always run the same, regardless of the environment it is running in. So that's what we use to encapsulate the computing environment. This is what we use to enhance reproducibility. We started using Docker containers in about 2014, 2015. Back then, it wasn't that popular, but right now, it is the standard in the bioinformatics community and beyond as well, many other communities. So our project is called BioDepot, and we have a GitHub page, and And you can see we have a really cool logo designed by students. I am not that good. So this project, I have already acknowledged Wes and Hong, and these are their photos on the screen. So now, any questions before I go on?
So will the containers increase the memory consumption?
It's very lightweight. It's a lightweight virtual machine. So you can think about it that way. And so we work on many different types of projects, but generally our projects falls into one of these three themes. One of them is we develop software to facilitate reproducible research. We make it easier for other people to reproduce their results. And we do a lot of data science collaborations. This is the part that I really love because I get to work with people who are different from myself and could have impact in many different areas. And the third direction is what we are currently working on a lot is a holistic optimization of computationally intensive tasks. So we try to optimize, make these computationally tasks run more efficient. We reduce the running time. If we reduce the running time, it would save us money on the cloud. And we do these types of optimization from 4 different angles. We optimize the algorithms. We optimize the software implementation. We optimize the way we deploy these tasks. And we also optimize the architecture when we deploy these tasks on the cloud. So we are going to touch on a little bit of these. So first of all, the first direction is developing easy-to-use tools to facilitate reproducible research. So here I'm trying to give an overview of a lot of work that we have done over the years. And these photos are our students, all graduates of UW Tacoma, who have been champions of these projects. As I said before, this is a recognition for the team effort for a lot of students. And so the first paper out there, Guldock using Docker containers with a common graphics user interface. So this work was published in 2016, and the champion for this work is Daniel Cristianto. Actually, he received the Husky 100 when he graduated from UW Tacoma. So in this work, what we did was we tried to add a graphical user interface on top of Docker. So Docker is wonderful for people who like to write code, but it's not so wonderful for people who don't want to write code. So we are trying to make it have a graphical user interface. And in this particular paper, we used a tool called X11 to do the rendering. And in the second work, we use what we call a Guldock VNC. VNC stands for the Virtual Network Computing Protocol. So by using VNC, we are creating a graphical desktop sharing system so that you can control the Docker containers from your browser. So that you create essentially a desktop inside your browser and you can click on it. So, and the champion for this work is Varun Mittal. And yet another work is we are creating an app to make it more automatically generate Docker containers, because Docker containers is not so easy to generate. You need to learn how to write the lines to create these Docker images. So we are trying to make it easier for people, so you could add certain libraries and we provide a template. So we create an app so that when you put it together with data science notebooks that we call Jupyter notebooks, it would make it easier to reproduce the work together with the Jupyter notebook. And by doing that, you could also automatically deploy your notebook on Google Cloud as well. So we have this work published in the Journal of the American Medical Informatics Association. And we have many champions of this work. We have Rim in this photo, and we have Jiaming Hu, we have Abir, we have Yash. They are all students from the MSCSS program. And the work that just got published a couple of weeks ago, building containerized workflows using the BioDepot workflow builder. So I would talk about this in a little bit more detail. That was published on September 11th in Cell Systems. And the champion of this work, the champion student is Jiaming, who has participated in so many projects for us. So BWB, it's an acronym for Biodepot Workflow Builder. So this is how it looks like. So what we do is this work builds on some other work that we have done. We again use VNC so that we are creating, you can think about a graphical desktop inside your browser. So all it is a container inside containers. So after you launch the container, you can look at it in the, interact with it in your browser. And so what we are looking at, all these icons, what we call, each of these icons, we call widget. Each widget represents one computational task. And each of these widgets use a Docker container to enhance reproducibility. So the idea is a drag and drop system, is you could drag a widget back in and you can drag a new widget on what we call the canvas to build the workflow. So it's modular in the sense that the widgets can come in or you can drag in a new one, and we use containers for each widget, in each widget. And yes, so it's all drag and drop, and it is amazing. So some of the other key features of BWB is, first of all, it's accessible. So the workflows would auto-install regardless of the execution platform. So you can run it on your laptop, you can run it on the cloud. It has a graphical user interface designed for biomedical users. So it's drag and drop, point and click, and you can deploy on your local computer or the cloud. Flexible, so you could export these, what we call pipelines or workflows, the entire sequence of computational tasks into shell scripts that you can run outside BWB, outside our system. And you could import in your own code, your own software to build new widgets using your own software. And you could do the mix and match, the drag in and drag out that I was describing. If we have time, we could even do a demo. Yes, I am very courageous to do a live demo. If not, we can watch a YouTube video. So it's also reproducible. It's not affected by software upgrades because we put in a lot of effort to ensure we explicitly specify which version we have been using in each of the pipeline. So we solve the dependency issue. And then it runs identically on any platform. And what is special about what we do, we also have the integration with many other tools that are used in the biomedical communities, like the Jupyter Notebooks, Cytoscape. And we also support optimized workflows that leverage, well, that will be, it's not yet. And we would also help you to manage cloud computing costs. So these are pending features. So we have a new story recently in the Tacoma page. So that is, if you want to find out more, to read the story, that would give you a quick couple of pages introduction to our work. And again, it's open source, it's supported by federal dollars, so it's publicly available on GitHub. You can, and the README menu is very, very comprehensive. And we have very cool YouTube videos that you could watch that are all linked on from our GitHub page. So that is about the software development effort. So in terms of the data science collaboration, as I said before, we love crowdsourcing challenges. And we get when we work on these crowdsourcing challenges in the classroom, we could also get publication opportunity in high impact journals. For example, in Nature Communications, that the class TCSS 588 is that class that I talked about that we got a publication out of the classroom project. And we also have other data science collaborations in terms of inferring gene networks. So the idea of gene networks is that given all these big, complex biomedical data, we want to figure out the relationships between different biological entities. So think about if we have different biological entities, we want to draw a line between two of them if they have a certain relationship. So this is what we are doing. We are developing the types of computational methods to integrate all these to get information from a different type of big biomedical data sources. So that's an example work that we published recently. And this work was just, this paper was just accepted last week or this week is we are working with Dr. Pamela Becker in hematology in UW Seattle about working on analyzing the drug sensitivity data generated in her lab. The idea is we adopt a personalized medicine approach so that we would be able to predict, to figure out what drugs would be more likely to work on certain patients. So, and the student here is Nicole Cower. She is a current MSCSS student in our school. So we are writing the full paper right now. So some more of the types of work we do in terms of optimizing computationally intensive tasks. So so one of the ways we are trying to optimize is the way we develop these containers, the way we deploy the containers. So in the first paper out there is what we come up with something called hot start containers. Is that so some of these computational tasks has an initialization step that every time when you run the entire job, the initialization step would be repeated again and again. So in this paper, what our idea is, we want to save the initialization step on the disk so that next time when you deploy the container, you can skip the initialization step, and that would save you time. So that's the idea in the first paper. And the next one is a paper that published, I think, in March this year in Bioinformatics. So what we have done is we take a workflow, what we call RNA-seq, RNA sequencing. It's one type of big data. So this workflow is in collaboration with Mount Sinai in New York. So they have a workflow that would run 29 hours if we just deploy on a standard virtual machines on AWS, on Amazon Web Services. So what we have done, we optimized it down to 3.5 hours by improving the algorithms and implementation. And then we are not done yet. So, and then recently, being inspired by Wes, we are looking into serverless computing, some new tools that are available in many different major cloud vendors. So we have this paper that Xingji, one of our students, just presented over the summer in Niagara Falls in Buffalo, is using serverless computing to improve the performance when we compare a lot of pairwise protein sequences. And yet another work we do is we are using serverless computing for RNA-seq data analysis. So the same workflow I talked about in the paper that was published in Bioinformatics 2019, that we took from 29 hours to 3.5 hours, so that we published that 3.5-hour paper. And now we can cut it down to 18 minutes. So now at this point, you must want to know what the heck is serverless computing. So let's do this. So the idea of serverless computing is a simplified programming model that we don't need to worry about configuring the servers, the machines. So you can think about in cloud computing, what we are doing is we are renting machines that sit somewhere else. And we pay by the amount of time that we are renting that machine. But the idea of serverless computing is we are not renting machines. We just send them the code that we need to run. And what happens is that they would charge us by the amount of time that we are running the job. That not running by the time that we have the machine running. So that is the difference. So you don't worry about machines. They configure the machine for you. They scale up or scale down for you. And all we pay for is the amount of time that the jobs are being run. And right now, it is very popular, and serverless tools are available on most major cloud vendors. For example, on Amazon Web Services, it's called Lambda. On Microsoft Azure, they call the Azure functions. On Google, it's called Google Functions. So you might ask, well, if serverless is so wonderful, why don't we all use serverless for everything? It's not so easy. They make it difficult for us. So for example, let's take an example for AWS Lambda. So they have quite a bit of limitations. We are limited by the amount of disk space, the amount of runtime, and the amount of memory, and the number of virtual CPUs on these Lambda. So in other words, what we are trying to say is that serverless is good for short-running, stateless, and event-driven applications. It's not good for everything. But what we want to do is, the question we ask is, can we adapt these bioinformatics workflows to leverage on these serverless platforms so that we could get you can look at them as a poor man's high performance computer, right? You don't need a cluster anymore. We just have many, many of these little computers that do a small chunk of the work. And we want to figure out what can we do about our workflow? How can we optimize our workflow so we can leverage on these new tools that are many, we can think about zillions of computers that are running everywhere that we cannot see, we cannot touch. So now, before we talk about how we did that, I would like to talk about the case study that we use for this particular workflow. This is this workflow that started at 29 hours and then 3.5 and now down to 18. So this particular data is in collaboration with Dr. Eric Sobey at Mount Sinai in New York. So what we do is they generate a lot of what we call RNA sequencing data. So they are looking at the effect of drug perturbation on many different cell lines. And so this is their type of experiments. So they have many different experiments. They have the controls, and they have some certain set of 24 drugs that they are interested in. And they also have some other set of drugs that they are trying to see to compare the impact. But for us, we are not so much worried about the inference of the results. We are more interested in speeding up this workflow, making it more efficient. So what we have done is we take this workflow and we have developed a hybrid architecture to run it on leveraging serverless tools. So we have the input data is we have some sort of files, a format we call FASTQ files. You can think about FASTQ files as sequences. These are text files that it has millions of sequences on these big files. And these files are about 46 gigabytes big and about 640 million reads. So a read is a short sequence. You can think about it that way. And so these are the input that initially sit in this S3 bucket. So S3 is a type of storage on the cloud, on AWS. So we have these 46 gig of data up there. And first of all, why we call it a hybrid architecture is we use the virtual machines, those computers we rent, as well as these serverless tools. So what we use is we first use a virtual machine. These computers we rent, we call EC2 instance, that we take these big data sets and we chop them up into smaller chunks. And after we have the smaller chunks, we put it back into the S3, the storage bucket. And from there, we launch many, 878 Lambda functions. We have a lot of these tiny computers. Each of these tiny computers are doing processing, performing the task on a smaller set of the sequences. And then when it's done, it's coming back to the S3 bucket. And then we do some merging and we do some counting. We put all the results together using yet another virtual machine instance. So what happens, the net effect is the entire workflow that originally required 29 hours is now can be done in 18 minutes. I believe the latest result is 15 if we turn the knob a little bit. And the total cost of the analysis is under $3. So at $3, you can save more than 28 hours. So this is a recap. So these are the types of projects we work on in the group. And as I said before, this is very much a team effort. And we have a long list of people to thank. And also, I would like to thank the funding agencies, National Institutes of Health. The acknowledgement for NIH is in green for a reason, because they give us money. And also thank collaborators at Madigan and at Mount Sinai, and also Amazon and Google for giving us the credits, many cloud credits to make this work possible. So this is where I want to end my presentation. So I started my presentation with cool photos of the team at UW Tacoma. And I want to end the presentation with even cooler photos of something else that, like of undergraduate research workshop. That diversifying the bioinformatics workforce is one of my many, many different passions. And I have the privilege to work with many undergraduate students in the area. So we recruit students across different majors in different years of their study at UW Tacoma and in the local colleges. And we have been doing since 2016, 2017, 2018, and 2019. So every year we have between 20 to 30 students that participate in our workshop. It has been a wonderful experience to work with these students. Okay, so thank you very much for coming today, and it means a lot to me for you guys to come, and have a good weekend. Thank you.